Hi Jen, hi Yvonne. The question I have
Hello.
for Hi. The question I have for you from our customers today is, what is your top tip for single cell dilution? Jen, would you like to go first?
Sure. So I think the first thing is exactly how dilute do you want it to be, right? So we all know that some cells, they need cell buddies in there with them. So some cell lines you can dilute down to, say, if you're doing a 384 well format, to one well or one cell per well. But you can also try different seeding densities. You could try 2 cells, you could try 5 cells, and see if that helps them be a little bit happier.
Yeah, and because you can still recover clonal populations sometimes from that, because they're just happier and they die off. Yeah.
Correct. You can always take that back and dilute again, right?
True, true, true.
But initially, you just need them to grow, and if that sometimes takes some contact with other cells.
So, Yvonne,
What
do
do you
you have
think,
any?
Yvonne?
Well, one thing that also one needs to consider is sometimes, as Jen said, they really don't feel happy in being alone in one well. They feel very lonely. So to try to help with this, what you can do is to add some conditioned media. Conditioned media is just cell culture media that has been in culture with the cell for 48 hours. Then you just need to take this media, pass it through a 0.2 micrometer filter so that it's sterile, and then give the cells this media, 50% of this media. So that way the cells will think or they will feel they have like company somewhere and they will be more happy to grow. Yeah, and this way you could really have a better recovery of the clone of the single cell, and they will start uh, multiplying and growing, and it will be easier for them. This will
Okay.
be one tip to do it.
Okay, good. That was really good. And we have a protocol as well for 96 well plate and 384 well plate single cell dilution, right?
That's
Yeah, and that
correct.
has all and that has all of those tips and extra tricks in there as well, right?
And
Yes.
it's not very different if you're doing one or the other, except obviously you're going to have different volumes and different number of plates.
Yeah. Sounds good. Great.
Exactly. And just like checking and marking where are the promising clones and doing like this continuous looking at the
Mm,
cells and checking. This is important so that you have a better control of where are actually your clones growing and which ones are the most promising ones because they have like a nicer growth.
exactly. So they're easier to deal with later on. Thank you so much, guys. That was really helpful. Bye.
Bye
Bye.
bye.